So what is Pten? Pten is nothing but it is phosphate and tension analog, right? So this is what you have to understand, that phosphate and tensive analog are homolog. So this is one of the tumor suppressor gene. That means this suppresses the tumor. It plays a very important in that. But you got some examples if there is any problem with this Pten gene, right? Which is a tumor suppressor gene, right? We already discussed about this tumor suppressor gene, the fourth one here. If there is any problem with that, we can get a few cancer conditions. So let us understand the most common malignance which with the mutation of the Pten, and the most common here is we got a cancer of endometrium, right? Type 1 endometrium. You can see this cancer of endometrium here. Type 1, also known as KRAS, type 1 endometrium cancer, also known as KRAS, is the most common malignant which is associated with this Pten gene, right? And if you look at the second most lesion, it can be benign or malignant here. So, right, the second most common lesion with the Pten is, what is that? It is uh, pre-malignant, right? It's a pre-malignant conditions, and this is known as congenital atypical endometrial hyperplasia, right? This is hyperplasia. It is congenital, right? It is congenital atypical, right? That is what it's written, congenital atypical endometrial hyperplasia. This is a second most common lesion. If they ask you second most common malignancy, then we have to speak this is a CA breast. It is the first most common malignancy if the Pten is type 1 endometrial cancer. The second most common malignancy is CA breast. If they ask you second lesion, so lesion, the second most common, it is what? It is congenital atypical endometrial hyperplasia. And the next, CA colon can also be associated with the mutation of Pten, and CA prostate can also be in the only, you have to understand, CA prostate is only male cancer exclusively associated with this Pten. That means the CA prostate, if there is any cancer of prostate and it is an exclusively, right, it's the only male cancer, right, only male cancer exclusively associated with this Pten, right? And this can also cause gliomas here. Gliomas is nothing but the tumor of nervous system of the glial cells. And if there is any problem with the Pten mutation can also lead into CA thyroid here, a cancer of CA thyroid here, and can also cause CA pancreas, right? In the CA pancreas, there is something known as condition known as Cowden's syndrome. So Cowden's syndrome, it is an autosomal disease, right? Dominant disease characterized by non-cancerous tissues like growth like hematomas. It's a non-cancerous tumor growth. So these are the things you have to understand in a round figure. If you know about this, let us see how do we remember this. The first thing, let us go with above downwards. So above downwards, you can like this is a thyroid, CA thyroid, and you can see the CA breast, second most common. And if you look at the things here, what's happening, this is the pancreas, right? You can see the CA pancreas can be seen. And the first thing you have to understand, this is CNS, so it is a gliomas, right? Gliomas, CA thyroid, right? You can see the CA breast, you can see the CA pancreas, you can see the CA colon here, right? So if you look at these, let's go down bit down, and you can get this CA prostate also if it is a male. And if you look at this in the female, you have got a type 1 endometrial cancer. So these are the important points you have to understand regarding the cancer, the examples which are associated with this Pten, the mutation of tumor suppressor gene, right? So let us understand the next concept here. The next concept is there is something known as DNA repair gene. So what do you mean by DNA repair gene? So if you observe carefully here, if there is any problem in the DNA, suppose if this is a cell, you got a nucleus at this aspect and you've got a DNA here. If there is any problem with this DNA, that can lead to mutation, that can lead to cancerous conditions. So God has given us few conditions, few genes in our nucleus, so that they can repair the gene, or so they can repair the DNA here. So they have been divided into, you got something known as the dependent and the independent. You can look at these, the P53 ATM, right? These are DNA, you can say DNA damage sensors. So they have got that ability of sensing if there is any problem with the DNA damage. That means they sense, yes, there is a DNA damage, and they call someone for the repair. So if you look at this is what you have to understand. BRC1A, BRCA2, they are repair agents. They are DNA repair agents. They are independent. They are dependent on someone, and they are independent. So this is the basic difference you have to understand in this aspect. So before going in detail, I think we have to understand the stages of DNA repair. How can the DNA be repaired? The DNA can be repaired if there is any problem with the, suppose if this is a cell which there is a mutation, whenever it goes into the cycle here, cell cycle, it has to be blocked. So first you know that there are three checkpoints. One at the G1 to S phase, the next you have got G2 to M, and next you have got in this metaphase to anaphase. So if you look at the first thing, the cell cycle is arrested at the checkpoint. These checkpoints, they see whether the cell is properly aligned or not. If there is any problem, if there is a DNA damage, and these are the checkpoints which stop the cell for not entering into the cell cycle. The next thing you have to understand, the second step is, once they are stopped, they repair the DNA. Or if they cannot repair the DNA, they can go into the state of apoptosis. They can send the cell into the apoptosis where they are killed because the DNA can't be repaired in them. So let us understand the mechanism of action, how these P53 plays a very important role in the DNA repair genes. So right, let us understand the concepts here. The first thing you have to understand regarding the P53 here is the mechanism of action remains same at both checkpoints. You know that P53 are controlled. If you look at the P53 here, this P53, they look at the checkpoints, right? They have got the mechanism of action remains same at the both checkpoints. So if you look at, this is what the cell cycle, and this is G1 phase, this is S phase, this is G2 phase, this is M phase. This is one checkpoint, and this is other checkpoint, this is a third checkpoint. And this first checkpoint and the second checkpoint is controlled by this P53. So the action of the P50 at the first checkpoint and the action of the P53 at the second checkpoint is same. So that is what it's written, mechanism of action remains same at both checkpoints. So let us understand how this P53 gonna work, right? So let us understand, whenever there is a damage, right, whenever there is a damage to the DNA, this P53, it does three things. One is it stops the cell, that is cell cycle arrest. The second thing does is DNA repair, and the third thing does is apoptosis. So let us go with the first thing. How can this go with the P21 here? So let us understand the concept here. Whenever there is a cycle, whenever there is a DNA problem, right? So you can see this, the P53 is stimulated here, stimulated in the sense, you have to understand before that there is something known as, if you look at this, you got, if there is any DNA damage, right? So that activate this, this has got the ability of P53. If you look at this, the P53 is present in this aspect, right? If there is any damage, the P53, the damaged P50, the stress are activated by, they activate this P53 gene, right? And you have to understand that these P53 for the cell cycle, for the DNA repair, cell cycle arrest, DNA repair and apoptosis, it calls some guys. So for the cell cycle arrest, it calls P51. It calls P51, so that the P51 comes and it stops the cell cycle. So this is cell cycle arrest. And if you look at the other point, this P53 can repair the DNA by calling this growth arrest and DNA damage. That is known as, there is something known as GADD4S, right? So this P53 call this GADD, and it repairs the DNA. And you have to understand that the P53 can call backs, and these backs, it leads to the apoptosis of cell if the DNA can't be repaired. So this is the basic concept. You have to understand the cell cycle arrest, DNA repair, and these apoptosis. So these are the important points you have to understand here. So let us understand the next thing here. The next one here is, if you look at this, the P53 can activate this MDM2, and MDM2 inactivates this P53. So what is it trying to tell us? If you look at this concept here, you can see there is a link between this MDM2 and this P53. And this MDM2, this P53, they activate these MDM2 proteins, right? You can see this, it activates this, but it has got a negative action, that this MDM2, it has got a negative action, or it inhibits the P53. So that is how it is regulated actually, that is how it is regulated. So the P3 levels should go down. So this is what you have to understand, and this is all it's written in our notes. So let us understand the next concept here. The next concept is regarding this. There is something known as, right, so that is what it's written. This P53 has got a negative feedback with this MDM2, right? So just understand that, right? So let us understand the next thing here. There is something known as ATM gene, right? We got something known as ATM gene. This is also a DNA repair gene, which we have already, we finished with P53. Let's go with this ATM gene. So if you look at the ATM gene, the mechanism of action, it changes with the checkpoints. It changes with the checkpoints here. So the mechanism changes according to the checkpoints. That means you know that you got a checkpoints here. The first checkpoint is present between the G1 to S, and the second checkpoint is present between the G to M. The mechanism of action of these ATM gene for the DNA repair, it all depends on the checkpoints. If you look at the first checkpoint, it is P53 dependent, and if you look at the second checkpoint, this is P53 independent. It is P53 independent. So this is what you have to understand, that the first checkpoint you have got a P53 dependent. That means these ATM, they activate the P53. You can look at this, what happens in the first checkpoint. I have already told you, if there is any damage, the ATM is activated here. Now the ATM, they activate the P53. The P53 same, they call the P21 and they stop the cell cycle. So that is what the same mechanism happens, what we spoke about the P53. But the only difference here is, in the first step, the ATM activates the P53, right? So if you look at the second one is G2M, the ATM and the G2M, you have to understand this is what the P53 independent. So this is what you have to understand. So let us understand what's happening with this P53 independent, how can this how the action goes on here. So let us understand the basic concept here. The basic concept of this here is, this is what the G2 phase and this is what the GM phase. You know the basic concept, that the checkpoint is we got an B plus 1, right? This is a checkpoint here, right? Which B is not right. So let us understand what's happening with these second checkpoint, where I told you this is P53 independent. If you look at the point here, in the second checkpoint, the ATM, right, you got something known as CDC25 phosphatase, right? So the thing you have to understand here is, if you look at this, there is something known as cyclin dependent kinase 1 phosphate. It is inactive state, that converts into active state of phosphate dependent kinase in the presence of enzyme CDC25 phosphatase, right? This CDC25 phosphatase, that converts the inactive CDK1 to active CDK1. So this is the first thing you have to understand this. So the thing you have to understand at this point here is, if you look at this, right, for the moving into G2 to M phase, right, for the moving into G2 to M phase, you have something known as a B plus 1. That means you have got a cyclin B and a CDK1, right? This is G2M checkpoint. Now this enzyme plays a very important role for the formation of this active CDK1, because if you look at this CDC25 phosphatase, that converts the inactive, right, the inactive CDK1 phosphate to active CDK1. So this is what the active CDK1. So this is how the CDC25 phosphatase acts. So suppose if there is any problem, if I inactivate this enzyme, so now the inactive CDK cannot convert into active. So you have to understand that now there will be like cell cycle arrest, because of the inactivation. Now there is no formation of this one, the cell cannot move from G2 to M. So that is what it's written at this point here, that is what it's written here, this in cell cycle arrest, right, the inactivation of gene, right? This is what it's written, that the ATM has got that ability of inactivation. If you look at this, why are we I started speaking in the ATM is P53 independent. So how is P53 independent? This ATM gene, right, it inactivates the enzyme CDC25 phosphatase. So once it is inactivated, it cannot, this enzyme cannot form active form. So active form is needed for the conversion of G2 to M. So there is cell arrest. So this is how the cell is stopped here. This is how the independent mechanism happens. So right, so let us understand the next concept here. The next concept is, you got something regarding, we got a few important points here. There is something known as DNA repair. DNA repair can be done by BRCA1 and BRCA2 and this ATM. So this is what the complex is, right? This is what the complex is. So ATM, right, BRCA1, BRCA2, so that those go for the DNA repair. So let us understand the next thing here is this P53, it undergoes apoptosis. It undergoes apoptosis. That means if this is then P53, it calls the Bach's protein, so that leads to the apoptosis of cell. Cell is dead. So let us understand few important points here. The few important points are, they may ask the most chemosensitive. The most chemosensitive will be the S phase. If you look at the C, they put one more C here ulta and becomes S. This is S phase. If you look at the radiosensitive, the radiosensitive will be the first G to M phase, then comes the G2 phase, then comes the M phase. So this is what you have to understand, that the radiosensitive first is G to M phase, first best answer, right? Then comes the G phase. If you look at this, comes the G2 phase, then comes the M phase. And if you see the most radio resistant phase, that becomes the late S phase. If you look, this is the late S phase, late S phase. So this is what you have to understand. Do not confuse with the chemotherapy. They put an ulta C, it becomes S, and radio sensitive is all these three here, and radio resistant is late S phase, right? So these are the important points, guys, you have to understand here. And the next thing you have to understand, there is something known as ionizing radiation induced DNA damage. Then only ATM gene will come. That means you have to understand that we need some ionizing radiation. So ionizing radiation can be a particle, can be X-ray, can be gamma. When they damage the DNA, then only the ATM gene will come. If the DNA damage is not caused by the ionizing radiation, the ATM gene do not activate, right? They do not activate. So this is what you have to understand. So these are the basic concept you have to understand here. So let us understand the next point, the example of what are those? The example of radiation induced carcinogens. So we got some radiation induced carcinogen. That means when there is a radiation here, right, and normally we got some mechanisms in our body so that we won't, though there is radiations here, it is protected. But in some conditions here, due to some problem, because of the radiation, we are gonna get a cancer here. So let us understand the examples of radiation induced carcinogens. The first will be mismatched repair gene. Mismatched repair gene. Suppose if you look at the concept here, if this is the DNA here, right, this is the complement DNA. In the complement DNA, if this is the point where it cannot, it is a mismatch pair, right? Mismatch. So normally this mismatch is cut, and then this is the new DNA, it is formed. It's a normal mechanism. Suppose in our body, if there is a problem with these defect, problem in this repair, then you get a mismatch, then that forms a proteins, and that can lead to cancer. So that type of mismatch repair defect is seen in HNPCC, that is hereditary non-polyposis colorectal cancer, right? So this is the basic concept you have to understand. Under the mismatch repair gene, this HNPCC comes. And the next thing you have to understand, there is something known as nucleotide excision. Nucleotide excision, you know, the nucleotide is nothing but which has got a phosphate, it has got a sugar, and it has got a base. Suppose if this is the DNA here, and if there is a problem with these nucleotide, that means it has contains a base, sugar, and phosphate, normally it should be removed. If it is not removed because of some problem, then that can leads to you have got something known as xeroderma pigmentosa. Xeroderma pigmentosa. In these conditions, the patients are sensitive to light. So this is what you have to understand regarding the nucleoside excision. And the next thing you have to understand, there is something known as recombination defect. You know that the DNA undergoes a recombination here. If there is any problem with the recombination way, and that can lead to ataxia telangiectasia, then Bloom's syndrome, and Fanconi's anemia. So what is ataxia telangiectasia? Ataxia is nothing but loss of control of bodily movements, right? Telangiectasia is nothing but it's a dilatation of capillaries. If you see next, Bloom's syndrome, it is a person where you have got a short stature here. It can be precancerous conditions. If you look at, this is what the Fanconi's anemia, right, where there is a bone marrow depression here. So one thing you have to understand here, this is the Fanconi's anemia. It's not a Fanconi's syndrome here. It's not Fanconi's syndrome. It's a Fanconi's anemia. These ataxia telangiectasia, Bloom's syndrome, and the Fanconi's anemia, they come under this recombination repair defect. So we got few important points under that, under this ataxia telangiectasia. You know that ataxia is nothing but it is a loss of control of bodily movements. In the cerebral ataxia, it can be a cerebral ataxia. The symptoms and signs are here. The signs and symptoms of this ataxia telangiectasia, the first is cerebellar ataxia. That is the reason he is in a state of a loss of control of body movements. So next you have got telangiectasia. I told you telangiectasia is nothing but it's a dilatation of capillaries. Next, in such condition, there is a growth retardation, mental retardation that can predispose to cancer, that then they dispose to cancer. So these are the basic concepts you have to understand. In the next session, we'll be dealing about the other concepts. Thank you.